6-carbon molecule. At the end of Krebs, all of those carbons have been turned into CO2. We lose 2 of them here, going from pyruvate to acetyl-CoA, right? Because we have 2 molecules of pyruvate for every glucose. So we lose 2 CO2 in this step. 6 minus 2 means we have to lose 4 more. And then we go do 2 turns of the Krebs cycle for every glucose, because there's 2 pyruvates. So we're going to lose 2 more CO2s for every turn, but that's 4 more for a glucose. So 4 CO2 here plus 1 plus 2 here is 6. That's our carbon accounting. So we lost, after the Krebs cycle, we've lost all of our carbons in CO2.
After the cycle twice.
And all of the energy that was in glucose is now in the form of ATP, NADH, and FADH2. Some of the NADH and FAD is produced in glycolysis, but most of it is made in the Krebs cycle. Questions?
Do we count one cycle for every 3 carbons cycling off of pyruvate, or are we counting one cycle as the whole glucose being produced?
So this has to go 2 turns for every glucose. So it does 1 turn for every pyruvate, but there's 2 pyruvates per glucose. Okay. Questions on this slide before we move on? All right, so that is the Krebs cycle. And we have a few footnotes here, so let's do these. All right. At the end of the Krebs cycle, for every glucose, how many carbons from the original molecule have been oxidized all the way to CO2? So at the end of the Krebs cycle, that means including everything from the beginning of glycolysis all the way through the Krebs cycle. All right, last chance to click in. All right, so most of you, 66%, I wish it was 90 or 100, saying 6, right? So at the end of the Krebs cycle, all of the carbon that was present in glucose has been converted to CO2. And just to review, if you missed that, because enough people did, I just want to go back to it one more time. For every glucose, all of this happens twice, right? Because for every glucose, we have 2 pyruvate. So for every glucose, all of this process is going to happen 2 times. We're going to lose 2 CO2s here, and we're going to lose 4 here. 4 plus 2 is 6. By the end of the Krebs cycle, all of the carbons in glucose have become CO2. And all of the energy is in the form of NADH, ATP, and FADH2. Okay? So those are really important key points. All right, so here we have it in slide form. All 6 of the carbons at the end of the Krebs cycle have been converted to CO2. NADH and FADH2 contain most of the energy, and I said there's been a few ATPs formed. 2 are formed for every glucose going through Krebs, and 2 are formed in glycolysis. All right, so now we have all these high energy NADHs and FADH2s, and we have to somehow get that energy converted to ATP, because the cell uses ATP as its chemical currency. So the whole point of the next step, which is the electron transport chain, is to get energy out of NADH and FADH2. There's a couple study guide questions I wanted to quickly talk about. Let me see if it's cooled off out here, like literally. So the first question says, what is a decarboxylation reaction? When we talk about decarboxylation, what's happening? I hear taking off of a carbon as CO2. So decarboxylation is when you lose a carbon as a CO2, we call it a decarboxylation. And this is where do decarboxylations happen in carbohydrate metabolism? Account for all 6 carbons in glucose. okay? So we said we lose 2 CO2s in the bridge step, we lose the other 4 in the Krebs cycle, okay? And so you should be able to do this, and I don't expect you to draw and name all the intermediates, but it is very reasonable to be able to draw 6 balls, these are the carbons, and show me where you lose them. So you say that you're going to lose them in Krebs and that you're going to lose them in the bridge step. That's a reasonable expectation. All right, at the end of the Krebs cycle, glucose has been fully oxidized to carbon dioxide. What molecules now contain the energy that glucose had? 3 molecules. ATP, NADH, and NADH2. Okay, so these study guide questions we've been talking about them really in a lot of detail. Questions? Okay. All right. The electron transport chain, I want you to have the big picture and understand how it works. I think it's easy to get bogged down into all the terminology and names, but this is actually not that complicated. Basically what you have is a series of molecules. Some of them are proteins embedded in the membrane. Some of them are little shuttle molecules that go between these proteins. And they basically carry electrons from a high energy level to a low energy level. That's basically all that's happening. We're going to remember our stairs that I drew not that long ago? My person I shoved over into the Grand Canyon? The same idea here. We're putting the electrons at the top of the stairs, and we're going to take them through little steps where each step they lose a little bit more energy. And we're going to take that energy and use it to create a chemical gradient of protons. And we're going to use the energy and gradient to make ATP. It's really just kind of 3 main things that happen. So what I want to show you is that NADH puts some electrons here, and the electrons go stair step down, stair step down, stair step down. Each, along the way, they're giving up a little bit of energy that the cell is capturing, and ultimately these electrons end up on an oxygen molecule forming water. So we talk about oxygen in the electron transport chain being the ultimate electron acceptor. Each one of these molecules accepts the electrons, and then it shuttles them to another molecule which accepts the electrons. Then it shuttles them to another molecule which accepts them. But at some point they actually end up on their penultimate or final electron acceptor, right? And that would be oxygen. The final ultimate electron acceptor is oxygen in this case. So the oxygen doesn't give, the water molecules take them and they don't give them up. See how oxygen is gaining hydrogens? Biology. The oxygen is being reduced, right? It's gaining hydrogens, and the hydrogens have those electrons. Okay. So here are some pictures. This is not a perfect picture. I have a video that's kind of nice too. One of the things that I liked about this picture, and I kept it, is because this compares some eukaryotic mitochondria with the prokaryotic membrane to just show that these cytochromes are in the membrane. They're in the inner membrane of the mitochondria. They're in the cell membrane of the bacteria. Okay, which goes back to our endosymbiotic theory and the idea that these structures became mitochondria in our ancient prokaryotes. If you look close up at these membranes, you can see these molecules, these cytochromes, buried in the membrane. And here we have our NADH donating its electrons to becoming NADH2. And these electrons are going to travel down, cytochromes down, down, each time releasing a little energy, and ultimately they end up on an oxygen molecule forming water. As they give up a little bit of energy, the energy is used to take protons and pump it into the space between, in the case of bacteria, the cell wall and the cell membrane. In the case of mitochondria, into this inner membrane space. So what's happening is you end up with a pH change. You have lots of protons in this inner space, but not very many protons inside here. When you have things at high concentration, what do they want to do?
Diffuse.
They want to diffuse back to low concentration, right? And they'll do that spontaneously. So the cell is pumping these protons against the concentration gradient. It's concentrating, it's using the energy in these molecules to concentrate protons in these spaces. And what's going to happen is the cell is going to say, okay, protons, you can go back through the membrane, but you have to make ATP on your way. So it's got a molecule called an ATPase, or ATP synthase, depending on which book you read. And as the protons go back through, it it's a molecular motor. It causes it to spin, and it uses that energy to make ATP from ADP succinate phosphate. So there's a lot going on here. I think the next slide, I'm hoping is a video. Oh, it's another picture. Okay, I got a video too. Actually, let's do this. Let's do this, and then we'll have to go back to that. I hope this is working.
During glycolysis and the tricarboxylic acid cycle, oxidation of organic molecules results in production of reduced coenzymes such as NADH. These coenzymes transfer hydrogens to the electron transport chain, which is located in the bacterial cell membrane. A hydrogen consists of a proton and an electron. The electron transport chain consists of a series of special electron carrier proteins that shuttle electrons from NADH to a terminal electron acceptor, such as oxygen. Electrons enter the electron transport chain when NADH transfers its protons plus electrons to a membrane-embedded carrier protein. The electrons are sequentially carried along the electron transport chain, while the protons are shuttled to the outside of the membrane. Some of the electron carriers, such as coenzyme Q, accept a proton from the inside of the cell membrane as it accepts electrons. The proton is then transported through the membrane as electrons move down the chain. This increases the proton gradient across the membrane and enhances the proton motive force. During aerobic respiration, the last carrier protein transfers a pair of electrons to oxygen at the end of the electron transport chain, and water is formed. The enzyme ATP synthase utilizes the energy of the proton motive force to synthesize ATP. This enzyme allows protons to pass back into the cell and couples the energy released in this process to the phosphorylation of ADP to form ATP.
What do you think? So this is what you need to know what's in this video. I'm not expecting you to know beyond this, but I want you to conceptually be able to tell that story, which is that you have these high energy NADHs that are depositing their electrons at one end of this electron transport chain, that their energy as they get to lower and lower energy levels is being used to create this proton gradient, and that the energy that's inherent in that gradient then drives the synthesis of ATP. That's really, I mean, I think the electron transport chain mystifies people, but that's really all there is to it. It's really, you know, you could memorize all the names of things. I'm not sure it's all that helpful. That's the basics. So let's go back to this slide and talk about it. There's one more detail that's important to know that they didn't talk about in that video. So let's review. So here's our NADH. Remember we made a whole bunch of these, right, in Krebs cycle? And we had 2 that we made in glycolysis. So there's lots of energy here. Here's our NADH. It's dropping off its electrons. The electrons are traveling down to lower and lower energy levels. Cytochrome Q is a transporter, so it's going to kind of shuttle the electrons. Shuttles them to complex, shuttles them to complex 4. The electrons end up on oxygen. We make water. It's all good, right? One of the things that you should know about NADH is you get about 3 ATP for every NADH. So this has enough energy in it to drive the production of about 3 ATP molecules. FADH2 only drives the production of 2 ATP molecules. So let's talk really quickly about why that's the case. And I'm going to draw my stair steps again, but this time we won't kill any humans. Okay? So here's our NADH becoming NAD+, and it's going to put its electrons up here on the stairs. FADH2 puts its electrons here on the stairs. And this is NAD+, FAD, right? I think I got that right. So there's electrons here. You see that this is at a higher energy level than this? So an ATP gets made here, and here, and here in terms of the energy. So if you put it up at the higher energy level, you have enough to make 3 ATP. But if you start in the middle of the stairs at a lower energy, what are stairs is really these cytochromes, right? This is the higher energy level cytochrome than where FADH2 does. It's dropped its electrons off. But you can think of it like stair steps. So you get more energy out of an NADH than you get out of an FADH2. Because the electrons start at a higher energy level. Okay? Questions on that? I can tell this is your favorite subject. All right. So we get 3 ATP for every NADH. We get 2 ATP for every FADH2. So that's what this is showing here, that the NADH is entering at a higher point in this gradient. And I'm showing this with ATP, but you see that a whole bunch of protons get forced in up here? If you put your electrons in here, you miss all of this, right? So you get less of a concentration gradient formed, and less ATP formed. You can kind of think of each one of these sets of protons as being responsible for an ATP. So when you put your electrons at the higher energy level, you get enough protons to make 3 ATP in your gradient. If you put the NADH, put its electrons here, it only gets enough protons to make 2 ATP. Yeah? I didn't say it was easy, right? All right, this is a great video, and you should feel free to watch it a couple of times and see just the basics are here. This is also an amazing, amazing video, but it's quite a bit more complicated. So I'm not going to show it, but if you are having no problem with this and you kind of like to explore a little further, they've gone and done a beautiful job graphics. The animation is just fantastic. It's kind of worth watching just for the animation. But I'm not going to show it because I think it might increase your anxiety level. And the pictures are beautiful, but it goes into way more detail than you need to know. Okay. In aerobic respiration, that's what we've been talking about, right? Aerobic respiration involving oxygen. Oopsie. Oxygen is required to all right, last chance to click in. Okay, so this is, I think this is hard stuff. So remember where oxygen comes in at the end of the electron transport chain? And what's going to land on the oxygen? The electrons, right? So if we go back, so here's our electrons, here's our oxygen. You see how it's collecting electrons? Maybe what was confusing here is ETC. That stands for electron transport chain. So B is the correct answer. It's the final electron acceptor in the electron transport chain. If you missed that, is that helpful? I hope. All right. So oxygen doesn't move hydrogen ions across the membrane. It doesn't catalyze this conversion, and it has nothing to do with CO2. What it is it's the place those electrons go. It's the final acceptor of electrons. After they've gone through all of the stairs, the cytochromes, right? They're going to land on, I didn't show it here, but they're going to land on oxygen. Okay. Why does NADH generate 3 ATP while FADH generates only 2? It's not open.
It's closed.
Oh, because I didn't click the button. There we go. All right, last chance to click in. All right, so the good news is, most of you are getting that right. I also have a few that are abstaining from clicking. So I know that often people are a little stressed when that's happening. But when F80H2, it simply enters the electron transport chain at a lower energy level. And because of that, you get fewer protons in your gradient, and you make less ATP.
So because it has two hydrogens, my assumption would be that it would have more electrons to give up.
Yeah.
Why does it enter the electron transport chain later if it has more electrons?
Yeah, it turns out that it doesn't have more electrons to give up.
Okay.
So even though it's got two hydrogens, so just because you're losing hydrogen doesn't actually mean you're being oxidized. It's just that in biology, for the steps we're talking about, it happens to play that way.
Okay.
So FADH2 doesn't actually give up more electrons than NAD.
Okay.
And it's actually where it's giving them up that counts. It's giving them up at this lower level of energy. So it's basically a less potential energy on the stairs at this point in the staircase than up here at this point in the staircase. Okay. All right, so now, spend just a couple more minutes talking about chemiosmosis, which is the energy that's in the proton gradient. So here we have our protons. We pumped all these protons into this inner membrane space. In the case of the bacteria, sometimes it's referred to as the periplastic space, the space between the cell wall and the cell membrane. So we have all these protons, and they want to move from high concentration to low concentration. That's the principles of diffusion. When you allow these molecules to go across, that releases energy. But the cell doesn't want to just give that energy out, it wants to use that energy. So what it does is, as these protons go through the ATP synthase, it's going to cause this motor, it kind of shows you the spins, this molecular motor to spin, and that's going to result in the conversion of ADP to ATP. So basically you're forcing, it takes a lot of energy to make ATP. You get a lot of energy out when you break ATP into ADP and inorganic phosphate. So it'll take a lot of energy to force that phosphate onto the ADP. And that energy is coming from this proton gradient and the motion of this ATP synthase as the protons go through. So we're releasing protons across that protein, and we're coupling the energy that's released to the synthesis of ATP. So we're using this energy, just like your car uses the energy in gasoline to make the tires move, right? We're using the energy that's in this protein gradient to make chemical energy in the form of ATP. So we're converting this energy, this potential energy, into this chemical energy. All right, and we just kind of answered this, right? We said as electrons are passed down the electron transport chain, their energy is used to create a proton gradient, right? And the proton gradient contains potential energy. And then that potential energy is used to drive the synthesis of ATP using the enzyme that's called
Coenzyme Q.
ATP synthase, right? ATP synthase is the enzyme that makes ATP. Okay, so basically these study guide questions are coming right out of the lecture notes, right, in the material that we've been talking about. All right. So let's just kind of go talk about the big picture, and then we'll talk a little bit about fermentation. So glycolysis is the first step. We produced only 2 ATP per glucose. Glycolysis is very rapid. It's kind of like a Ferrari. So when cells want to make energy really fast, they'll use glycolysis. If you're running up the stairs to get to class on time, primarily what you do is you're burning sugar. Your body is taking glycogen from your muscles and burning through the sugar really fast. Actually, you go into oxygen debt because your body can't go through aerobic respiration fast enough to fuel your muscles. Someone who's running the 100-yard dash is using basically just glycolysis to generate energy. So it's like a Ferrari. It's super fast but really inefficient. Does this make sense? Really fast but really inefficient. When you have oxygen present and you can go through the Krebs cycle and electron transport chain, I got a Prius here. I'll should probably turn this into a leaf or something now. But this is actually slow but very efficient. So instead of only generating 2 ATP per glucose, in a eukaryotic cell you get 34 ATP molecules for the original molecule of glucose. And in a prokaryotic cell you actually get 38. Have you ever driven a Prius? When you step on the gas, nothing happens. For a few minutes, and then it catches up. I had one, I really liked it. But anyway, but it's slow, right? But it's very efficient, you get great gas mileage. So glycolysis is kind of like a Ferrari. In a situation where there's oxygen, you can actually make ATP faster, but not efficiently. The electron transport chain in Krebs, in the presence of oxygen, you can get a lot more energy out of a glucose molecule, but it's not very speedy. Questions on this? And then we're going to move on to fermentation. All right. Now, if you're struggling with today's material, but you didn't do what I suggested on last week, and practice the Krebs cycle at home over the weekend on a blank sheet of paper, then what I would suggest you do is go home and practice the Krebs cycle and electron transport chain on a blank sheet of paper. And then work on those study guide questions. And if you're still struggling and conceptually not getting it, then you should come see me in office hours so we can go over it together and make sure you understand it. Okay? I know it can be hard, and we've kind of broken it down into the pieces. And I think sometimes what makes this material hard, because the math isn't particularly hard, and the concepts, we're really focusing on the big picture ones, but I do think it's a little bit scary. People may have learned this before, and you've heard lots of terminology thrown around, but it's really something that's graspable. I think if you're going to, understand the complexities of giving people pharmaceuticals in a hospital environment when they're really sick, that's way harder than this. So you can do it, but it will require a little bit of practice. So if you're feeling lost, you're going to work on Krebs and electron transport chain on a blank sheet of paper. You're going to practice the study guide questions. And then when you have a good understanding of where you're stuck, you're going to come talk to me because I can unstick you. Okay? But I can't unstick you if you don't come talk to me. Okay. All right, what about beer? So some microbes don't need oxygen. In fact, we talked about the fact that some microbes are actually poisoned by oxygen. And they can use a process called fermentation to generate energy. And fermentation is really just glycolysis with an extra step. It's an alternate pathway to Krebs cycle and electron transport chain, and the cell is going to get its energy from glycolysis. So why do we do fermentation? It's got a really important job. All right, so fermentation allows us to produce energy without oxygen. You can use the same molecules. You don't use Krebs or electron transport chain. And instead of oxygen being the final electron acceptor, we're going to use an organic molecule instead. Remember I said it's fast, but it's not very efficient. So for a glucose molecule, you're only getting a little tiny bit of the energy out of it that's available. Do you undergo fermentation? Sort of, right? When you're going through, running up the stairs, you can make lactic acid, right? You don't do it long term. But some of your muscle cells can do it very short term. All right, so here's our process. Here's respiration that we've been talking about. Glycolysis forming pyruvic acid. And then we said after we make pyruvic acid, if there's oxygen present, we'll go through the Krebs cycle. And then all these NADHs and FADH2, the energy will be collected essentially in the electron transport chain, and we're going to turn it into tons of ATP, with oxygen being our final electron acceptor. If there's no oxygen, your muscles are starved for oxygen because you've been running, or if you're a bacterium in an anaerobic environment, you can undergo fermentation. Here's the problem. You see this NADH that's been produced here? If you just go to pyruvic acid, eventually this molecule builds up, and since you don't have oxygen to accept these electrons, eventually you run out of NAD+. Because you've used up all the NAD+ to form NADH. Does that make sense? Eventually you're going to run out of this molecule. NADH has to give up its electrons somehow. And it does it by producing molecules like ethanol and lactic acid in fermentation. So let's look at the chemistry for a moment. All right, and I just said this. So why ferment? We need to regenerate NAD+. So here's glycolysis, and for every glucose we make 2 NADHs, so we're using up NAD+ and we're making NADH. Eventually the cell is going to run out of NAD+. And when it runs out of NAD+, can it go through glycolysis? It needs NAD+. That's part of glycolysis. It's a reactant. Once you run out of NAD+, you're going to shut down glycolysis, and now even your Ferrari's not working, right? Because you can't go through and get ATP out of glucose without NAD+. So the cell had to come up with a way to recreate NAD+. And what it does is it takes pyruvic acid and it uses NADH to reduce pyruvic acid to something else. And so you see we're regenerating our NAD+ this way. And there are lots of different products of fermentation, but you're going to only have to learn 2 of them, the 2 most important ones. Okay? So fermentation, I'll show you at the end a whole bunch of ways that we can get byproducts from fermentation. But we're going to focus on lactic acid, which happens in some bacteria, but also in your muscle cells, and alcohol fermentation, because who would want to live without this one? All right. So here's our glycolysis. We've made pyruvic acid, and here's the 2 examples. So if we take pyruvic acid and we simply reduce it to lactic acid, we can actually undergo lactic acid fermentation. We regenerate NAD+, and it can go back to form more rounds of glycolysis, to allow more rounds of glycolysis to happen. That's pretty easy, right? If you look at pyruvate and now look at lactic acid, you see that this has more hydrogens on it than this molecule. So we have reduced pyruvic acid and we have reoxidized NADH to NAD+. The other option is we can take pyruvic acid, we can lose a carbon dioxide, which makes a molecule called acetaldehyde, and then we can take this molecule and we can reduce it to ethanol. In the process of reducing acetaldehyde to ethanol, we're going to oxidize NADH to NAD+. And now NAD+ is available to go back and allow us to do more rounds of glycolysis. So the whole point of fermentation is to make NAD+, so the cell can go back and continue to go through rounds of glycolysis. Question?
Yeah.
I answered it. Okay. That happens once in a while. All right, so I just got more pictures of kind of the same process here for you to look at. So here's lactic acid fermentation. Here's glycolysis, glucose going to pyruvate, right? And here's our pyruvate molecule, and we're going to convert it to lactic acid. See how this carbon is being reduced? And we're going to, because this gets reduced, we're oxidizing NADH to form NAD+, and now this can go back and participate in more rounds of glycolysis. Streptococcus, lactobacillus.